All right, Rebecca, are you ready? Can you hear us?
Yes.
Okay?
Yeah, ready to go. Can you see me?
I can. Looks good.
Great.
All right. Welcome everyone to our Hatfield Research Seminar for today. My name is Cinnamon and I am the Research Program Manager at Oregon State University's Hatfield Marine Science Center in Newport, Oregon. And I am your host for today. Just before we get started, a couple of logistics. Your mics, cameras, screen shares have been turned off. Help us out and keep it that way for now. If you'd like to participate, which we encourage, please use the chat bubble at the bottom. You're welcome to type in your questions at any time and we'll get to those at the end. Wanted to also let you know that this event is going to be recorded and will be posted on the Hatfield's past seminar page. So if you need to share it, you're welcome to do that. It'll be up in a couple of days, so give me a couple to get it on. And then I wanted to make a couple of announcements. Next week, believe it or not, is already July 2nd. We'll have another seminar with Dan O'Vander, who is a postdoc with the University of Washington, who'll be talking about assessing population level conservation effects of marine protected areas. So we hope that you come back and join us again next week, same time, same place. But the other thing that's happening, which we're really excited about, is we have an event this evening as well. It is our second virtual Science on Tap. That event will start at 6 p.m. tonight. Angie Dewar, who is the marine fisheries specialist with Oregon Sea Grant, will be talking about Newport's fishing fleet. So come join us, learn a little bit more about what happens here in Newport. And like I said, that event starts at 6 this evening. If you need any more information about any of our upcoming events, you need links, you need details, you can log on to Hatfield's homepage, scroll to the bottom, and there's a calendar of events there with all of those details. But for today, I would like to introduce today's speaker, which we're really excited to have join us. Rebecca Mostow is a PhD candidate and NSF graduate fellow with Oregon State University, where she works with Dr. Sally Hacker. She received her BA in biology from Orban College in 2013, where she worked on desert plants. A little bit of a change for her, working with us now. Before she started her graduate degree, Rebecca conducted research and taught at the Alaska Maritime National Wildlife Refuge, the Bureau of Land Management in Carson City District, and at the Port Townsend Marine Science Center, where I also spent many, many years before coming to Oregon. So it's nice to have her back in my life for sure. The work she's going to talk to us about today, she's earned awards and funding from the National Science Foundation, Washington Native Plant Society, and the Society for Integrative and Biology. And the work that she has been doing, part of it is set up at Hatfield currently between housing and our brand new building. So I'm really excited to learn a little bit more about what those big white bags are all about. So Rebecca, I'm going to hand it off to you.
Great, thanks, Cinnamon. Hi, everyone. Thank you all for virtually being here. And huge thanks to Cinnamon for inviting me to give this talk and setting it up. It seems like there's been a lot of great seminars happening recently. So I am thrilled to talk to you today about part of the dissertation that I'm working on here at Oregon State University and in the Department of Integrative Biology. So the talk that I'm going to give today is titled A Wild Grass Chase, Hybrid Beachgrass Discovery on the Pacific Northwest Dunes. So let's get into it. So the story I'm going to tell you today is really the story of a discovery that's been unfolding on the Oregon Coast Dunes for the past 8 years. And the arc of the story that I'm going to tell today is we're going to start with this cast of characters who are the people who are involved in this discovery. Then we'll get into some Pacific Northwest dune natural history, although maybe we should call it unnatural history, because this is most of the plants I'm going to be talking about today are actually not native to the Northwest. Then we'll get into the botany of desire. Don't come at me, Michael Pollan. I just mean hybridization. We're going to talk about plant hybridization and why it matters. And then we'll get into this unexpected discovery that I'm alluding to. And then at the very end, we'll talk about sort of the consequences and next steps. And that's when I'll tell you about those big white bags marring the view of the new Marine Studies building. All right, so before we go any farther, I do just want to take a quick poll. In normal life, I would have you raise your hand, but now we live in Zoom. So if you could just raise your Zoom hand, you can do that by going into the participants section. Raise your Zoom hand if you have read the book Dune. I'm gonna watch sort of the results roll in and just tell you a little bit more about Dune. The book is a novel all about this planet completely covered in sand dunes. And okay, there's a small number of people who have read this book. I am going to reduce the amount that I talk about Dune and the jokes that I do about it. But the quick aside is that, okay, more people have read Dune, good. Frank Herbert, who wrote this book all about sand dunes, this like sci-fi novel about a planet of dunes, he actually lived in Salem and visited the Oregon Coast dunes and became so inspired by them as this like weird, amazing system that he wrote a whole sci-fi book about them. So the ecosystem that I want to talk to you about today is fascinating. It's inspiring. It's definitely a little bit weird. Yeah, that's all I'll talk about Dune because it looks like most of you are not going to get my jokes. Okay, so moving on, let's get into the real story. So now we're in this cast of characters section. So the first person is me. I'm Rebecca Mostow. Cinnamon already really introduced me. So I'll just say, I am now a graduate student in integrative biology, working on my dissertation. I sort of came to IB through a sort of meandering path that took me through the Port Townsend Marine Science Center, where I spent a lot of time with my head in fish tanks, like you can see right here. But I also got the opportunity when I wasn't knee or shoulder deep in water, to interact with community or citizen scientists for the first time. And I really learned the value of citizen science and became super inspired by the idea of trying to incorporate citizen science into my work. So later in the talk, I'm going to talk to you about a community science initiative that I'm working on, but just shout out to the PTMSC and to that inspiration coming from there. Another really important character in this story is Dr. Sally Hacker. She's a professor at Oregon State in integrative biology. She's been studying these dunes for a long time, and she is my dissertation advisor. So I'm not going to make any jokes about her. Sally is here today. That's another good reason not to do any teasing. So thanks, Sally, for all your support in this project. And it's important to mention whenever we talk about scientific endeavors that science really does take a village, and the type of project that I'm going to talk to you about today with all these different pieces and parts, it could not be possible without a ton of different people. Some of them are on this slide right here, from undergrads who have helped me with data collection and data analysis to grad students who go out in the field with me. We sort of like pair up and team up to work on each other's projects, to professors from different universities. There's a couple people I just want to point out. One is Peter Ruggiero, who's right here, who's a professor in CEOS. And so anytime you hear me say anything about like dune shape, dune morphology, anything like that, that is work that's been done in collaboration with Peter's lab. So thanks to Peter. And then the other person is Felipe Barreto down here. Unfortunately, I don't have any funny field pictures of Felipe yet. You gotta come out in the field with me. But so Felipe is a professor in integrative biology. And so anytime you hear me say anything about genetics, genome size, anything like that, that's work that's been done in collaboration with Felipe. All right, we're gonna dive into this dune ecosystem natural history. And this is a slide showing you sort of what the natural native state of the Pacific Northwest coastal dunes really is like. What I want you to notice is that black and white photo in the background has these low, rolling, hummocky dunelets. There's lots of bare, open sand. There's vegetation, but it's sort of clustered in these little bits, and then there's really lots of open areas. This is a really diverse habitat. I'm highlighting some of the like beautiful, incredible native plants that are present in sort of a natural dune system in the Northwest coast. So it's super diverse, really dynamic, constantly shifting sand, and sort of sparsely vegetated. Now, this is what the dunes on the Oregon and Washington coast looked like until around the early 1900s. And then more people started to move out to the coast and try to settle on the coastline. And they started to introduce beach grasses. And beach grasses are groups of grasses that can sort of be planted on the dunes and grow really densely and start to grow and build and stabilize dunes. So now, after that beach grass introduction, the dunes that we see on the Oregon coast look more like this, right? They are these tall, linear, super stable, really densely vegetated dunes with pretty low plant diversity, right? You're really just seeing these grasses out there and not very much else. So that's the transition that we have seen on the Oregon and Washington coasts. And just to give you a sense of the scale that I'm talking about, of the scale of the change, here's some overhead photos of Pacific City. So on the left, we have these sort of pre or early introduction photos. And then on the right, you have some of the later post beachgrass introduction photos. And what I want you to notice is just how different they look. On the left, you're seeing tons of this like white open sand. That's all this like that open sandy natural dune habitat. And then on the right, you start to see these sort of really densely vegetated green areas here, these linear foredunes along the coast that are not present in the older pictures. And not only are these dunes and these sandy areas densely vegetated, but they're also densely populated. So like, look at the extensive community and human development that's happened on these dunes. So that's really one of the main reasons that it's possible, is because of these beach grasses that were introduced. So the spread of the beach grasses, the planting of beach grasses on the Oregon and Washington coast had some really significant positive effects. They have done exactly what they were introduced to do. They have increased coastal protection. So this picture is illustrating sort of the dunes as almost like a seawall in front of those houses, right? It's protecting those houses from storm surge, sea level rise, like all of those high wave events, all that stuff. And the dunes have also increased the stability of the sand on those beaches. So there's less likely to have sand just blowing onto the houses that are right behind those dunes. But the beach grasses have spread outside of the areas where they were initially planted. So they were initially planted just in areas where there was human development, but now they've spread into more natural areas. And they have also had some really negative effects. So the beach grass spread has decreased native plant species richness by sort of outcompeting the native dune plants. And it's also decreased the habitat for the western snowy plover, which is threatened. And I'm showing the pink sand verbena, which is a state listed endangered plant here too. So there have been some really significant positive consequences for sort of the human development of the coastline because of beach grass introduction. But there have also been some very significant negative consequences of the introduction of these beach grasses in terms of biodiversity management and species conservation. So I've been talking about the beach grass introduction as sort of one thing, but really what I haven't mentioned is that there's actually two species of beach grasses that were introduced to the Pacific Northwest. One of those species is Ammophila arenaria, and that grass was introduced first, and it was primarily introduced in Oregon and California. And then the other species is Ammophila breviligulata. That grass started to be introduced a little bit later, and it was primarily introduced in Washington. So these two grasses have slightly different ranges now. They look really similar, but they actually have differential effects on dune morphology, so the shape of the dunes, coastal protection, the value of those dunes for protecting communities built in their lee, and biodiversity. So they have different effects on the plant species richness on the dunes. So for the next part of the talk, we're going to sort of zoom in on these two species and think about what the differences between them really are. So the two species are Ammophila arenaria, European beachgrass. That beachgrass is native to Western Europe and was introduced to the Northwest. It's not native here. And then Ammophila breviligulata, American beachgrass, which is native to the US Northeast and Midwest. And I just want to take just a moment to point out these confusing common names. So Ammophila breviligulata, the common name is American beachgrass, which often leads people to think that maybe it is native to this part of America, but it's not. So don't get confused. It's native to the East Coast, and that common name is confusing, so we're going to stick with the Latin name. So Ammophila breviligulata, native to the US East Coast, invasive here. So these two grasses look really similar. They occur in these sandy coastal environments. But one really good way to tell them apart is to look at their ligules. So a ligule is the piece of tissue between the grass leaf and the stem. It's this little membranous piece of tissue that I'm highlighting here. In Ammophila arenaria, it's like really long and pointy. And then in Ammophila breviligulata, it's really short and flat. And you might notice if you're looking at that Latin name, right? Ammophila breviligulata. Brevi means short, ligulata means liguled. And so the Latin name really says this is the short liguled Ammophila. So if you're trying to remember the difference, check out the Latin name. All right, so let's look at the relationship between these grasses and the different types of dunes that they build, the different effects they have on their ecosystems. So Ammophila arenaria dominated dunes tend to be taller and steeper than those dunes built by Ammophila breviligulata, which tend to be shorter and shallower. And that is because Ammophila arenaria grows more densely. So the stems tend to be more closely clustered together, and there's more vertical growth and less horizontal growth in Ammophila arenaria. So that growth form feeds back into the shape of the dune. And then Ammophila breviligulata tends to build these lower, shallower, less densely vegetated dunes, and that is because it has more lateral growth form. The two species also have differential effects on the plant species richness of the dunes that they dominate. So Ammophila arenaria dunes tend to have higher plant species richness than those dominated by Ammophila breviligulata. And what I just summarized is work from Sally and our lab in the early 2010s. So there's another important beach grass that you might see out on the coast, and that's actually our native grass. So this is Lamus mollis, or American dune grass. Once again, these common names are confusing. So this is American dune grass. This is a native plant. American beach grass is an invasive plant. It's too confusing. This is Lamus mollis. It's native to Oregon and to the Pacific Northwest. And it's a really beautiful plant. I always say that, and then I think maybe I'm the only person, or maybe only a dune ecologist would think that this grass is beautiful, but I find it quite lovely. It's got these thick stems. It has sort of a blue-green tint to it. And this is the grass that builds sort of the native dune system dunes, those like really hummocky, low rolling dunes. And that is because the stems don't grow very closely together. They grow really well spread out. Dunes dominated by Lamus mollis have really high plant species richness. So remember that first slide I showed you with all of those beautiful native plants. Those are the dunes that Lamus mollis builds. And Lamus mollis dunes have something that the beachgrass dunes don't have, which is they have high habitat value. So there's our little western snowy plover happily nesting in the little like swales in between those hummocks in that bare sand area. So all of the things that I've just summarized for you, they were discovered by our lab mostly through the use of these long-term monitoring surveys. So the lab has a long-term monitoring project that returns every 3 years to all these sites along the Oregon and Washington coast. So the sites are shown as triangles in this map here from a lab mate of mine, Ruben Beal. So every triangle is a site, and then at each site, when we return every 3 years, we survey the shape of the dunes and the plants that are growing on it. So we do that by running a transect perpendicular to the shoreline over sort of the profile of the dune. And at that transect, we assess the dune shape. We do that. Here's Katya doing just that. This is my lab mate Katya with this backpack GPS on. No, she's not communicating with aliens, which is actually what we get asked about all the time. We're out here. She's measuring the shape of the dune. So that GPS tells her not just where she is in space, but sort of her altitude, how high she is. And from that, we can get the profile of the dune. And then we also do these plant community surveys where we lay down a transect and quadrats that look like this. And in the quadrat, we count all the stems and identify all the plant species that are in there. And from those two data sets, when we compare them, we can find correlations between the plants, the grasses that are growing on the dunes and the density of those grasses and the shape of the dune overall. So it was actually during one of these surveys in 2012 that my lab mate, Ruben Beal, encountered a plant that just kind of looked weird. It had these thick stems like Ammophila breviligulata, but it had a longish ligule, almost like Ammophila arenaria. And it was the only plant on the whole dune that was flowering. And so he looked at this plant and he thought, could this be a hybrid? What's going on here? And so that question, could this weird plant be a hybrid? That is really the question that has spurred a big chunk of my dissertation work. And that's what I'm going to be getting into today. So before we go too far, we'll just define hybridization. We're really talking about interspecific hybridization. So a hybrid is the offspring between parents from two different species. So no, not this. More like this. So a mule is a great example of a hybrid that people are familiar with. So a mule is a cross between a horse and a donkey. Post Tiger King mania, you might be comfortable with this one too down at the bottom. A liger is a cross between a lion and a tiger. But there are also lots of common food that are hybrids that you might encounter in the grocery store. So like a pluot or a tangelo, those are both plant hybrids. And these are all artificial hybrids, so hybrids produced by humans on purpose. But there are also tons of natural hybrids. Hybridization in the natural world is not uncommon. I've thrown up some examples from animals, warblers, salamanders, but then there are also even more examples of hybridization in plants. So I have some really common examples of plant hybridization, sort of model systems of plant hybridization. Sunflowers, irises. So those groups are sort of known for being willing to hybridize and sort of that hybridization is quite common in their systems. And another group of plants that is really well known for hybridization are many groups of grasses. And so when we're asking this question, could this weird plant that Ruben found on the dune, could this be a hybrid? We need to think a little bit more closely about the species that we're dealing with. So here are some things we know about the plants that might favor hybridization in this system. In their native ranges, both of the Ammophila species hybridize with a different Calamagrostis species. So we know that both of these Ammophila species can do hybridize in their native range, but there has never been an Ammophila arenaria, Ammophila breviligulata cross recorded. However, that probably has to do with the fact that this range overlap, this area where these two grasses co-occur in the Pacific Northwest, it's really the only place in the world where they co-occur at this kind of density. A couple other factors favoring hybridization are they have the same number of chromosomes, so they're sort of genetically compatible in that way at least, and they overlap in their range, and they overlap in their phonology. Their flowering times overlap just a bit. So here is a map showing the range overlap between the two Ammophila species. We have Ammophila breviligulata in black up here. You'll see that I'm going to use this species code AMBR to talk about Ammophila breviligulata a lot in this talk, and then AMAR is the species code for Ammophila arenaria. I'll point it out again, but just in case. So Ammophila breviligulata is the dominant grass in Washington, but it actually extends all the way down to Pacific City. And then Ammophila arenaria is the dominant grass in Oregon, but it extends actually all the way through Washington. So there's this fairly large range overlap between the two species. And not only do they co-occur at that sort of large regional spatial scale, they also co-occur at a local scale. So here's Ammophila arenaria and Ammophila breviligulata growing right next to each other on the dune. And you might even notice that Ammophila arenaria has that really dense growth form that I was talking about earlier. And the two species, not only do they grow next to each other, we found that they sometimes flower next to each other. So here's Ammophila arenaria flowering stalks right next to an Ammophila breviligulata flowering stalk. All right, so I set out to investigate this potential hybrid and asking the question, like, is this weird plant that Ruben found on the dunes, is this a hybrid? And the things that I looked into was I started by looking at the morphology of the plant. What does it look like? How does that compare to the parent? Then I moved on to the phonology. When does this weird plant flower? And does it flower at a different time than the parent species? I looked at the genomes of the plants and estimated their genome size and compared that. We did a little bit of SNP genotyping. And finally, I'm looking at the range. Where is this sort of weird potential hybrid and how much of it is really out there? So for the next part of the talk, we're going to go through each of these questions, and I'm going to show you sort of how I did these investigations and what the results look like. So the first thing I did was look at the morphology, and we did this by collecting hundreds of plants and measuring a wide suite of traits on those plants of both the hybrid and both Ammophila parent species. Some of the traits we measured are just really important for being able to identify the plants, like ligule length and leaf width. Those are just about being able to identify them. But some of the traits we measured are really important for dune building and dune building potential. So the plant height, questions about how dense it is, those are really important for what kind of dune is the hybrid going to build. And what we found is that the hybrid is intermediate between its parents in some traits. It has an intermediate length ligule, as I'm showing here in the picture. It has an intermediate number of shoots or stems coming out of each plant. It has an intermediate blade width, and it exceeds its parents in some traits. It's taller than its parents. It has heavier below ground biomass. So I'm going to show you, zoom in a little bit on some of the data from this project, just a subset of the data. So what I'm showing here is comparisons of the morphology by species. The x-axis is what species we're talking about. Again, AMAR, that's AMAR, HYBR is the hybrid, and then AMBR is Ammophila breviligulata. So that's the x-axis, and then the y-axis is going to be the trait that I measured. So here's the graph for ligule length, and you can see Ammophila arenaria, this really long ligule, Ammophila breviligulata, a really, really short ligule, and the hybrid has this intermediate length ligule. Here's the graph for leaf width. So again, the hybrid is intermediate between its parent species. And here's the graph for shoot height. So here's a trait for which we found the hybrid actually exceeded its parents. This is the height from the base of the plant to the very tip. And finally, we looked at the number of shoots, the number of stems coming out of a single rhizome. And you can see the hybrid is intermediate between its parents, but it actually is not statistically different from Ammophila breviligulata. All right, so the next move was to dive into the phonology of the plants. And so here what I'm looking at is the flowering time. And so what I did to assess this was I returned to a number of sites over the course of an entire flowering season. And at each of those sites, I counted the number of flowering stems that I found. And so here is the data from that part of the analysis. So the x-axis is the date that I did the survey, and then the y-axis is the number of flowering stalks. And here I'm just showing you the data from Ammophila arenaria. So each of these little dots is a survey that I did, and what you can see is that Ammophila arenaria sort of starts flowering around mid-June, and the flowering peaks around the end of June. So if we went out on the coast right now, we would probably see a lot of Ammophila arenaria flowering. Okay, I'm going to overlay the Ammophila breviligulata flowering. So here we are. As you can see, Ammophila breviligulata flowers later than Ammophila arenaria. It's peaking around late August, and it's really just starting to flower now. If we went out on the dunes, we might find a couple flowers, but not likely. Okay, now I'm going to add the flowering time of the hybrid on top of this. And here's what you can see. So the hybrid has an intermediate peak of its flowering time. Its peak flowering is around mid-July, and it overlaps with both of the parent species. So this is an exciting piece of data for me. What I see here is the two parent species overlap a little bit in flowering time. So there's this small area where there's this small period of time where both parent species have flowers open on the dunes where they could potentially be hybridizing. And the hybrid overlaps with both of its parent species, but has a different peak than either of them, indicating that it could potentially backcross. If it was able to backcross or reproduce sexually with its parents, it could potentially do so on the dunes. One note about the magnitude of the graph is just that I have not controlled for the density on the dunes, so that's why the peak of Ammophila breviligulata looks so much higher. There's just more of it at the sites. All right, so moving on to the genome size. So I estimated the genome size of a number of individuals from each species and from the hybrid. Then this work was done using Ryan Contreras's lab in horticulture at Oregon State. And this is just flow cytometry data. So what I'm showing here is that Ammophila arenaria, we found that it has a smaller genome size. Ammophila breviligulata has a larger genome size. And the hybrid has an almost exactly intermediate genome size. It has a genome size that looks almost exactly like it has half of its DNA from one parent and half from the other. This is pretty compelling evidence that what we're looking at is a first generation cross. So an individual that really was made by a cross from Ammophila arenaria and Ammophila breviligulata, they produced this hybrid. It's sort of the first generation hybridization event. So to verify that that's what we're seeing, I did some SNP genotyping. And so here's the results of that. I'm not going to go too deep into the methods that I used here, but what I'll tell you is that we did a reduced representation genome library building, and then we isolated some SNPs from that, and then I fed those into a computer program called Structure, and Structure assigns each individual sample into a group, and so each column in this graph is a sample, and then the color is what group Structure wants to put it in. And so on the left we have Ammophila arenaria. Structure is coloring it all red, and then on the right is Ammophila breviligulata. All of those columns are getting colored blue. So Structure is saying these are two different groups, two different populations, and here is what the hybrid looks like. So each of those columns are getting placed sort of half in the Ammophila breviligulata group, half in the Ammophila arenaria group. So this really does look like what we're seeing is a first generation hybrid cross between these two species. All right, so the next part of this project is to look at the range of this hybrid. So we have some really compelling evidence that what we're looking at is a first generation hybrid cross, and now we need to know how much of it is out there and where is it. So we're going to zoom in on that range overlap that I mentioned before. And so we're zooming in on an area between around Pacific City, Oregon, all the way up to like Ocean Shores, Washington. And on the left, I'm showing the 4 sites that actually Ruben Beal found. So he initially discovered the hybrid at one site on the Long Beach Peninsula, and then he found it at a couple other places where he was like, I'm seeing the same sort of weird looking plant. So 4 sites found between 2012 and 2014. Then between 2017 and 2018, I joined the lab and I started to really investigate this. One of the sites up in Grays Harbor, that red X, that actually eroded, which is what I get for working in coastal systems. So that site is no longer there, but we did find a couple other new sites during that time frame. And then between 2018 and 2019, I did a more extensive survey of the northern Washington coast and of the Olympic Peninsula, and I found a number of more sites. So this is what we're looking at now. Here are all the known sites where we've found the hybrid. So this is around 12 sites where this hybrid has been found, and they range all the way from Ocean Shores, Ocean City area down to Pacific City. And you might notice that there's some sort of gaps in this map, right? So Long Beach, it seems like there's some holes there where you might expect to see more hybrids. Grays Harbor, there's definitely some holes. And then all of that Oregon coastline, we might think that the hybrid would be there, but we haven't found it. And the truth is that really those are just areas that I haven't looked very closely yet. That area up here around Ocean Shores, I looked very carefully at all these beaches and I found a lot of the hybrid. So this map is showing me that I really need to look more, spend more time on the beach surveying for this hybrid so that we can get a sense of how much of it is out there and where it is. Okay, yeah, and here's just some labels of where those sites are. Grays Harbor, Long Beach, and then there's this cluster of sites in the Fort Stevens area where we found a number of hybrid patches. So going back to this list, my ways that I was going to investigate the hybrid, I looked at the morphology, and yes, the hybrid looks different from the parent species, and we can quantify that difference. Then I looked at the phonology, and yes, the hybrid flowers at a different time from the parent species, and it overlaps in its flowering with both parents. Then we looked at the genome size and found that the hybrid has a genome that's intermediate in size between both parents. We did some SNP genotyping and really got some very significant evidence that this is a first-generation hybrid. And finally, we're investigating the range of the hybrid, but we really need more data to narrow down on, to zoom in on sort of how much of the hybrid is out there and where is it. So when I add all of that together, what I see is that I come to this sort of exciting and unexpected conclusion, which is that the Ammophila beachgrasses are hybridizing in the Pacific Northwest coast. And that discovery comes with a number of pretty significant potential consequences. So the first one is about coastal protection. So remember from the beginning of the talk, I talked about how these beachgrasses build the dunes that protect coastal communities in Oregon and Washington. So we don't know what kind of dune this hybrid could build, but it could potentially change the shape of those dunes and affect coastal protection all along the coast. There are also some very significant consequences for biodiversity conservation. So the hybrid, we know that the parent species negatively impact our native flora and fauna, and we don't yet know whether the hybrid will perhaps exclude more native plants than those parent species do or build better habitat. We just don't know what the consequences on biodiversity will be. There's also a potential consequence for the population genetics of the parent species. So this hybrid could potentially backcross with its parent species. And you can imagine, for example, if the hybrid repeatedly backcrosses with Ammophila arenaria, that could be an avenue for Ammophila breviligulata genes to flow into Ammophila arenaria. So we could get some gene flow between the two species, mixing that has potentially never been seen before and could change the invasion dynamics of this ecosystem. So population genetic diversity is closely related to the invasive potential of plants. And so any change in population genetic diversity could change this invasion ecology on our coast. And the other thing that could change the invasion ecology on the coast is just the hybrid itself. So hybridization is closely tied with invasive ability in plants. And so there's a potential that the hybrid itself could be very invasive and could spread and compete with the parent species, changing the invasion dynamics here. And that's really a question about the ecological interactions with the parent species. So how is the hybrid interacting with its parent species when it's growing next to them? Is it competing with them? Is it facilitating with them? And how is that sort of changing the ecosystem, the sort of invasion ecology on the coast overall? So this is kind of a long list of serious potential consequences that I think we need to keep our eyes on. And so to sort of dig into those consequences, I have more questions. Science at its best is iterative, right? So this like one discovery has led to a number of different questions. And these are just three that I'm gonna highlight here today. But one of them is really how much of the hybrid is out there. And although this seems like a basic question, it's like really central to our understanding of those potential consequences. To assess how the hybrid is going to affect dune shape, we need to know where it is so that we can look at the shape of those dunes. To understand how it's interacting with its parents, we need to know where it is. To understand its effect on biodiversity, we need to know where it is. So this question, this sort of basic how much of it is out there question is really at the heart of a lot of what we're doing here and understanding those consequences. Another question is, how is the hybrid interacting with its parent species? Is it facilitating them? Is it competing with them? What's going on there? And then a third question is, how will the hybrid affect the dune shape and habitat value? So to investigate these questions, here are the methods that I'm using. So for that first question, how much of the hybrid is out there, I'm launching a community science project or a citizen science project. To investigate how the hybrid is interacting with the parent species, I'm doing a common garden experiment. And to understand how the hybrid is affecting dune shape and habitat value, we really need to do more dune surveys. So go to sites where the hybrid is and look at what those sites look like. And actually the lab is bringing in another graduate student to tackle that question because it's so big. So for the last chunk of the talk, I'm going to spend a little bit of time, basically an aside, talking about this common garden experiment because it's actually set up at the Hatfield Marine Science Center. So some of you may have seen it already. And if you haven't, then you'll see it whenever you get a chance to go back there. And then the end of the talk, I'm going to focus on the community science project because really the reason that I'm giving this talk is I'm really hopeful that some of you might want to be involved in the community science mapping project. All right, so here's my aside about the common garden experiment. So the question we're asking is, how does the hybrid beachgrass interact with the parent beachgrass species? And to do that, what I did is I planted these bags filled with sand with different combinations of plants, so different species planted together, and also different densities of plants. And this is what it looks like. There's the cool new Marine Studies building in the background, and here are my big white bags. This is, so the Marine Studies building is there, and then on the other side of this picture are all the dorms. So these are the bags. They're all of the different combinations of plants planted together. And the idea is that these are like mini dunes, and we're competing the different species against one another. We'll let them grow together in sort of a common garden, so sharing the same space and with the same environmental conditions for 2 full growing seasons. So for all of this summer and then all of next summer. And then next fall, I'll come back and rip all these bags open, take all the plants out and dry them. And then I'll start to look at sort of who was the winner of the competitions. So for example, is there a species of grass that grew much bigger or has more stems or is much healthier, even when it's growing with the other species. So that's sort of the idea of the project. And here's an overview actually taken by Mark from looking down from the Marine Studies building. So it's a pretty cool shot. This is what you'll get to see when you walk around on the ramp, is my little project down here. And so the setup is that there's 9 treatments of different densities and different combinations of plants. And then there's 6 replicate blocks. And then there's 1 very tired grad student measuring all of those plants. I think I'm 2/3 of the way through measuring grasses in 54 bags right there. So I'm pretty tired. And a huge thank you to the Markham Award. That's actually one of the main reasons that I'm able to do this project, and to a Sea Grant to Sally and Felipe Barreto. Without that funding, this project would not have been possible or would have been at a very, very, very small scale. So thank you so much for that. All right, so the last chunk of the talk, I'm going to talk about this community science project that I'm so excited about. So the goal of this project is to survey beaches for the Ammophila hybrid with the help of community scientists. And I'm doing this project in collaboration with the Oregon Shores Conservation Coalition, which houses Coast Watch. And the project itself is being housed on iNaturalist. So before we go any further, I guess I'll do another quick sort of poll. Could you raise your Zoom hand if you have used iNaturalist before? I just want to judge people's sort of comfort with iNaturalist before I dive too deep into it. Okay, there's some results coming in. Cool. Okay, so I'll just give you a little bit of a background about iNaturalist and how it works. iNaturalist is a social network, and the goals of iNaturalist are to connect people to nature and to generate scientifically valuable biodiversity data. It just so happens those are my goals too. So we're well matched, me and iNaturalist. So the way iNaturalist works, if you haven't had a chance to use it before, which it looks like a number of you have, which is great. The way it works is basically individual people submit observations of the living things that they encounter in their life. So basically you go outside, you see a bird, you take a picture of that bird, and you upload that picture onto iNaturalist as an observation. So every observation has a time and date stamp and a location and a photo usually, and a tentative identification. So you say I think this is a robin or whatever. Then you upload that onto iNaturalist, and then the community part, the social network part, is that every other user can see your observation and they can agree or disagree with your identification. So they can be like, yes, that's a robin, check, or no, that's a towhee. And so it's really like a learning network where you sort of learn together to identify the organisms that you encounter in your world. So that's sort of the social part, but the biodiversity data part is pretty incredible. This is just a screen grab of iNaturalist. If you put in Ammophila, or marram grass is the common name that iNaturalist uses for these plants. That's the European common name. And so what you're seeing here is basically a range map. So each of those dots is an individual observation. And when we have thousands and thousands of dots, we end up with something that's not just an individual, but is really like this beautiful range map that we're seeing. So the Ammophila beach grasses are present in the Northwest. Here's our non-native populations here. And then over here, this is the US East Coast and Midwest. This is the native population of Ammophila breviligulata. And over here is the native population of Ammophila arenaria. So it's a pretty cool setup. And the way that iNaturalist is housing this project is that I built a project page within iNaturalist that just aggregates all of the observations of these beach grasses, including of the hybrid, that are made on our coast. So the project page is just pulling in everything that gets identified and gathering it all together and making basically a range map like that for us. But it also has a lot of information if you want to be involved. So it has an about section, sort of a description of the project. And it also has this identification guide that I made. So the ID guide has a chunk, a sort of block for each of the different species. And then it also has some identification tips. So it reminds you to sort of check the ligule, check where you are. And then it just tells you at the end, sort of like, if you can't identify the beach grass, that's okay. You should just submit it under the label genus Ammophila. It will automatically get pulled into that project page and I will see it and I can help identify it. So if you're unsure of your ID, take some of those close-up pictures and you can still submit it and we can learn together, right? I'll identify the plant for you, you'll get a notification that I did, and you'll sort of learn how to identify them better. Just a quick aside, if you don't want to use iNaturalist, but you really do want to help with this project, great. That's awesome. Just email me your photos. I will put them on iNaturalist. You don't have to like get involved with this tool if you don't want to. So if you would like to help, if you would like to be involved in this project, I would love your help. And the first step would be to just check out that iNaturalist project page. If you go into iNaturalist and you go to the project section and you type in hybrid beach grass, the page will just come up. I also have the link at the end of this presentation. And then get into iNaturalist and practice submitting an observation. Just observe anything in your natural world, practice doing it. And then the next time you're out on the beach, try to identify the beach grasses that you see. And if you really want to get more involved, I'd love to have you. I'm going to do a webinar training on Monday at 11. And you can just email me for details about that training. I'll put my email on the very last slide. And we would love to have you come sort of get more involved. And in the training, I'm going to talk more about how to identify the grasses and where to look, all of the sort of nitty-gritty details. So with that, I just want to say thanks so much to all of you for being here. I'm pretty thrilled with the turnout today. And a huge thank you to all of the different people who were involved in this project. I showed you their pictures on that slide early on, but there's been a team that has worked on this research project as advisors and co-authors, a team who has been out in the field with me, a team that has done grass measurements. This research was conducted on the traditional homelands of the Clallam, Makah, Quili, Chinook, Quinault, Chehalis, Lower Chinook, Tillamook, Siletz, and Nistika people. So there's a huge debt of gratitude owed there. And then this project received funding from the Oregon Sea Grant. That's a grant to Sally and Felipe, an NSF graduate fellowship, and the Markham Award, which has been really instrumental for me. So thank you all for coming to this talk. And I think we have some time to do some questions, so we can open it up.
Wonderful. Thank you so much, Rebecca. That was wonderful. Go ahead and type in any questions you might have in the chat box as we kind of get started here. And as we give people just a chance to do that, Rebecca, I had a question for you about when you were showing us the maps of the presence of the hybrid, that was kind of a presence absence kind of snapshot. I was wondering about the extent at each of those and whether that seems to be changing or spreading. And I don't know if you can speak to that or not.
Yeah, that's such a great question. And actually, so I mentioned, I kind of had a little aside that Sally is bringing in another graduate student to help on this project. And one of the things that she's going to look into is sort of, is the hybrid spreading or not? At most of those sites that I highlighted, what we're seeing is just a single patch of grass. So the grass is spread underground with these underground stems. And so we're seeing just these individual patches. But because of just the large scale of that map, there's actually many more patches than were showing up on that map. There's one site that we found, there's actually two sites we found where the grasses are much more spread out. It's not just a single dense patch, but it's really like sort of the grasses, the hybrids are growing intermixed with the parent species. And those sites are both in Oregon, but every other site is really just like discrete patches.
So we're getting a couple of comments. Most of them are just, yay, good job. And a couple folks that are saying they're interested in helping, so they'll contact you that way. I have a question here. Did you notice any difference in the rodent species across the different grasses or any other types of animal interaction in those different groups?
Wonderful question. Thank you, Timothy. One thing that I have done in the dunes is to do a little bit of tracking in the back dune and have had a really fun time. I don't see any differences in the mammals that we find in the back dune, but that's a great question. And I know there has been some research actually in California, Timothy, about the presence of native and non-native rodents in these heavily infested dunes. But that's not different by species, just by invaded or not.
Nice.
Thanks.
Any other questions? Feel free to put them in the chat. And again, this is just me and my spatial brain. I can ask another question. The hybrid, do you see it on the foredune and the back dune as well? Is it along the transect, or is it specific to kind of a location in that dunescape?
Super great question. Actually, I'm very into this question. So the hybrid has been found predominantly at the toe of the dune. So at the part of the dune that's closest to the water. There are a number of things that could explain that. One of them is that that's where I'm most likely to find it, right? If I'm walking along the beach and I see something weird, the place that I'm most likely to see something a little bit strange and take a look at it is the toe of the dune, right? I'm not walking in the crest of the super dense dunes, right? But there are some other possible, like actual ecological or biological reasons why the hybrid might actually just be at the toe. And my favorite of those is that the grasses flower more at the toe, which is cool to see. They actually like have a really high rate of flowering at the toe, and it's much more than is at the crest of the dune. This is, you can see, so like this is Rylea, which is weird, but there's this area where there's super, super dense flowering and then basically no flowering in the back of the dune. And that might have to do with the fact that the grasses really like to have sand deposited on them, and they grow better and they're just more healthy in areas where the sand is getting deposited. Or it might have to do with the fact that they're younger when they're at the toe of the dune. So great question. We're finding it mostly at the toe of the dune. I have some theories about that, but it might just be luck.
More time, more data, we'll answer more questions. Okay, we have a whole bunch of questions coming in. Is there any way to determine how long the grasses have been hybridizing?
That's a really good question, Mike. What we do know is that the first patch that was found in 2012, and it was already pretty large. So there's kind of an idea in the back of my head about could we figure out the growth rate of that patch and then sort of back calculate when it was first formed. Sally and I have talked about trying to do that with some of the plants. Another thing is that dunes are sort of this, I just alluded to this a little bit, but dunes are a really classic system for studying space for time. What is that called? Space for time. Basically this ecological idea that there's parts of the ecosystem that are younger and then parts of the ecosystem that are getting older. So the toe of the dune is the youngest part of the dune. And so if we're finding hybrids there, maybe we can figure out like how old is the toe of the dune and then figure out how old the hybrids are. If we're finding hybrids at the back of the dune, maybe we can figure out how old is the back of the dune. So those are just some ideas. I also have some ideas about sort of like genetic clock stuff, but honestly, yeah, I'm less sure about those. And
there's a couple of genetics folks on this call, so maybe they have some suggestions as well.
Yeah.
Okay, so another question is, can you guess what might happen as far as beach management or restoration for plovers if the hybrid increases stability?
Yeah, that's a really good question, and one that we just really don't have the answer to yet, but it's one of the most important reasons why we need to figure out, is this hybrid spreading, and what is its effect on the shape of the dunes and sort of the density of the plants. One possibility, we have a little bit of evidence that it sometimes grows very, very densely. And so that would be really bad for plovers. They do not like a super dense dune system, a densely vegetated dune system. But other than that, we just really don't know. And so we need to investigate more because the beach grasses have such a negative impact on those plovers.
So we have another question about the highest plant diversity and the difference in the different species. Do you know the mechanism on what's driving the difference in the diversity of the native plants in those different types of grasses?
We don't for sure know the mechanism. So I think the question is about, so Ammophila arenaria has a higher plant species diversity on its dunes than Ammophila breviligulata. One possibility is that research out of our lab has shown that Ammophila breviligulata is actually a really, really strong competitor. So it outcompetes Ammophila arenaria when you put it in a bag experiment, like I showed you. It outcompetes pretty dramatically. And so one possibility is that Ammophila breviligulata is a very strong competitor against most plant species, and it's just sort of outcompeting them. I think that's kind of the most compelling idea. The other possibility, right, is that there are differences in their ranges. So maybe the Ammophila arenaria range has different sort of physical conditions that might make there be more open spaces or something like that helps the native plants.
And that question came from Annette. She also said that she wanted more thoughts on dune succession into forest, but she'll talk to you via email.
Yeah, we should talk about that.
Nice. And the question right below that, can you see that, Rebecca? I'm not exactly sure. Is a hunt in the Oregon dunes, Florence to Coos Bay, continued worthwhile at this time?
A hunt for the hybrid is, I think, what the question is asking. Like, should we be looking for the hybrid there? And I would say, if you can get up to some of the North Coast beaches, that would be a better place to spend your time. But if you're down in the South Coast, in the Coos Bay, Oregon dunes, Florence area, and you're interested in learning to identify those grasses, there's always a chance you'll find something weird. And so it's always worth just looking, but the probability that you'll find the hybrid is very low because Ammophila breviligulata is really not down there except in like a couple of very weird places where it was introduced.
Nice. But absence of data is still good data, so go with that. It looks like that's the end of the questions that we have right now. I highly suggest anybody that is interested contact Rebecca directly, maybe participate in the training on Monday. And so, looks like there's one more question I might have missed.
Oh yeah, I see one about the Canadian range.
Great. If you can see it, will you read it out loud? Others can hear it.
Yeah, I got a question. Are you interested in the Canadian range for Ammophila arenaria, Ammophila breviligulata, and Lamus mollis? And if we ever see the hybrid? Absolutely. I'm super interested in that. The reason that sort of the maps end in Oregon and in Washington is really just, that's just a political boundary. Like the plants that boundary doesn't affect them. And actually we know that Ammophila arenaria and breviligulata occur pretty far north into Canada. The farthest north site that I know about right now is in Haida Gwaii on the northeast coast of Haida Gwaii. So that's sort of the extent of the range. But yeah, I think a really interesting question is, are there areas in Canada where these two species co-occur? Because if there are, then the hybrid could be there.
Well, that's great. There's a whole bunch more. Just thank you so much for coming in, and nice job. And yeah, what an amazing project. And we're just excited to have you be at Hatfield and to watch what happens in the next 2 years.
Great.
So, for everybody online, thank you so much for joining us. If it's possible, join us for our Science on Tap this evening, possibly next week. And know that this will be recorded and posted on our page, and you can share it for anybody who might have missed it and wants to learn more before the webinar on Monday. And Rebecca, thank you again so very much for joining us.
Yeah, thank you all for being here, and thanks Cinnamon for inviting me. And if you see me out by those bags, you should wave.
Perfect. All right, everybody. We'll sign off for now, and thank you so much.
Bye, everyone. All
right, I'm going to end the meeting. Thanks very much, everyone.
Yeah, thank you.